Welcome to Brain Ponderings, your window in the scientific world of brain research. And today we're going to talk about a topic that we haven't talked about before. It's a cell type that, next to neurons, are probably the most interesting type of cell in the brain. And these are the oligodendrocytes. They insulate. They're best known for like insulating, nourishing neurons, axons, but they have other roles and they have a very interesting biology and brain development, injury, disease. And my guest today is an expert on oligodendrocytes. It's Professor Richard Liu from University of Cincinnati. He's the Beatrice Lamkin Endowed Chair in Cancer Epigenetics. And the second half of our conversation, I'll have him talk about cancer because he's doing a lot of work on that, and you'll see why it's relevant to glial cells. He's also scientific director of the Brain Tumor Center and professor in the Department of Pediatrics. So he has a background in developmental neuroscience, which myself, I do too, actually. Okay, Richard, welcome.
Hi, Mark. Thank you so much for having me. It is my great honor to have this opportunity to talk with you. And I have greatly enjoyed and also learned a lot from your Brain Pondering series.
Oh, that's great. Let's talk about your background. So you're from China originally?
Yes. So I was born in the southern part of China. Yeah. So, and then I moved to Beijing for my undergraduate study, Beijing Normal University. Right.
Yeah.
Yeah.
And then you came to the US and you went to New Jersey?
Yes, I went to New Jersey, Rutgers University.
Yeah.
Yeah, so I was study under Dr. Masayori Inouye for my PhD advisor. So I don't know whether you know him. Masayori is a pioneer in bacterial gene regulation, RNA biology, and also protein engineering, right? So yeah, he actually recently got indicted, become a member of the National Academy of Sciences, right?
Yeah, I don't know him personally, but I know the name and kind of the general what he's best known for. So initially you're getting a nitty-gritty molecular biology and getting a good foundation that can enable you to study any cell type and look at it in a critical manner, manipulating genes, for example, or interrogating signaling pathways, et cetera, et cetera. So then,
Exactly.
did you get interested in neuroscience then?
Yes. So actually, as you mentioned, right? So I learned a lot from the Dr. Inouye, right? So particularly in the molecular biology and molecular cloning. So I think at that time I think I can do a lot, outside different fields, right? So I was interested in neurobiology, particularly for cancer research. So because my Dr. Inouye has a wonderful motto is challenging the unknown. So that has been inspire me for my whole career.
Yeah, that's great. And then your postdoc is when you really took off and really started to, well, it was up in at Harvard, right?
Right. So yeah, so I joined Dr. Chuck Stout's lab at the Dana-Farber Cancer Institute, Harvard Medical School. So Chuck, his seminal work is on the link between the oncogene with the growth factor signaling.
Yeah.
And so, yeah, particularly for those, he discovered a lot of immediate early gene at that time, so which would respond to the growth factor signaling, right?
Right. So obviously growth factors promote the growth of cells and in a lot of cases division. For example, in the brain, stem cells in the hippocampus that a lot of people have studied, we've studied a little, there's a growth factor called fibroblast growth factor 2, FGF2, that promotes proliferation. And
Yes.
then it's been known for a long time that in a lot of cancers, these even even in the absence of a growth factor, that the pathway activated by the growth factor can be active. So it's like out of control. It's like continuous excessive activation, right?
Exactly. Right. Yeah. Particularly recently, in many of childhood tumors, so you have those growth factor receptor fused with other gene, so therefore they can get constantly activated
Yeah.
without growth factor stimulation. So that actually can lead to cancer formation.
And that has
Mm.
has led to some treatments for cancer, or
Yeah. Yeah. For example, track fusion.
uh,
So people have been using the track inhibitor to block
yeah,
the signaling, and also FGF receptor signaling fused with other gene like TACC 1, 2, and 3. So actually, that actually can lead to continuously activation of FGF receptor signaling.
yeah.
Right.
All right, and then
Mm.
okay, so you're up at Dana-Farber in Boston, and you're in a lab working on cancer. And then what about oligodendrocytes, like
Yes.
their
So
normal development and so on?
yeah, I think that's a very interesting question. So at that time, we were interested in gliomagenesis, and we believe the glioma are coming from glial cells.
Right.
So, but once and then we hypothesize if we can identify those genes that control glial cell differentiation or proliferation, we can control tumor growth.
Yeah.
But to my surprise, so I came from outside the field. I found out nobody actually knows about the glial specification genes. So which is in stark contrast with those in the neurogenesis. At that time, people have identified many of the neurogenic factors, but nobody actually identified any of the gliogenic factors. Yeah, so I think they even have one hypothesis at that time. They say if a neural stem cell cannot form cannot form neurons, they will stay as glial cells. So you don't need anything to push them to form glial cells.
One of my colleagues, so when I was at NIH at the NIA, soon after I got there, I recruited Mahendra Rao. You must have come across him.
Yes, right.
Yeah. And so he's a good friend of mine, and I learned a lot from him about the genes controlling not only oligodendrocytes, but neural stem cells. Right. So first, talk about oligodendrocytes.
Mm-hmm.
They come from stem cells, and then talk about what the normal function of these cells is.
Yeah. So because we at that time we don't know the glial specification gene, so therefore I set a screen to culture neural stem cell and then push them and treat them with the cure for glial formation, and then we perform the gene differential gene expression to try to find out those genes potentially control glial cell differentiation. So at that time there was no microarray or RNA sequencing, right? So we have to perform the screen by manually, mainly through those degenerated oligonucleotide primed PCR.
Huh.
Probably you used that before. So just target specific transcription factor family, for example, basic helix to helix transcription factor family, and homeodomain protein family, and zinc finger protein families. So yeah, I was lucky enough to identify a pair of basic helix to helix transcription factor. We name it as oligo 1, 2. So and the reason we named them as oligo 1, 2 because we find these two genes has a very unique expression, particularly in oligodendrocytes. So that's why we start my career, right, to work on the oligodendrocyte lineage progressions, right.
Okay, so you have these stem cells, the oligodendrocyte progenitor cells, or OPCs.
Mm-hmm.
They can divide and keep proliferating, or when they get the right signals, they can stop dividing and differentiate into these oligodendrocytes, which then they interact with axons,
Mm-hmm.
and it's very intriguing. They actually keep wrapping around the axon till you have all these big layers of essentially lipid, because it's membrane, which is mostly lipid. And so the lipid, it doesn't conduct electricity very, it's kind of like a wire, right, being insulated. And so
Yes.
these are arranged, one oligodendrocyte will wrap around a portion of the axon, then there'll be a gap, and another one will wrap around, then a gap. And so when a neuron fires, the electrical charge essentially jumps across that gap. And so this dramatically speeds up the conduction velocity. And this is critical for communication of neurons that extend long ways within the brain, from the brain to the spinal cord, from the spinal cord to muscle in your big toe. And
Yes.
but there's been a lot of discoveries on other functions of these oligodendrocytes. They're doing much more than just insulating. There's a lot of communication.
Yes, yes. So the oligodendrocyte is different from the Schwann cell in the peripheral nervous system. So when they become myelinated, they can myelinate multiple axons.
Ah,
So yeah. So the Schwann cell only myelinate one Schwann cell with one axon, but oligodendrocyte can do multiple axons.
ah.
And I think this in the field people believe so there's like a activity dependent or independent of the myelination process by
Oh,
oligodendrocytes, right?
yeah.
So and then recently there's a lot of study actually beyond those myelin formation by oligodendrocytes. And so the oligodendrocyte can actually, particularly for the oligodendrocyte precursor cell, they have those they can actually connect with the neurons. So they express synapse so that they can connect with neuron and then modulate the neuronal activities.
And is that through classic neurotransmitters or some other signaling
Yes,
mechanism?
still they still can. They have those AMPA receptor in the oligodendrocyte precursor cell we call
I see.
OPC, right? So yeah, still goes through the classical neurotransmitter signaling, right? So and
Oh,
also
glutamate.
glutamate too,
I
right?
don't think I put that in my book on glutamate. There's,
Yes, so their study actually indicate that the glutamate receptor in
yeah,
OPC can respond to the neuron activities. Yes,
yeah, yeah,
yeah,
that's very important.
right.
The glial cells, they also, in my lab, did a lot of work on brain energy metabolism, and particularly like intermittent fasting, exercise effects on the brain. And these glial cells, there's been a lot done with astrocytes playing some role in energy metabolism, but there are some data indicating that the oligodendrocytes play an important role in the energy metabolism in the axon, because these axons, they have to have a lot of ATP, so they have a lot of there's mitochondria all along them,
Mm-hmm.
and so that's another interesting function.
Yes, so the recent study actually showed the oligodendrocyte can produce the lactate, so lactate can be uptake by the axon for
Yeah,
the energy homeostasis,
yeah.
and so I think you talk about the intermittent fasting. I think it's very interesting. So there's a recent paper just published a few days ago. They actually show there's a beneficial of the intermittent calorie restriction for the patient with MS.
Oh,
So,
yeah, well, yeah. So at Hopkins, so actually Ellen Mowry at Hopkins, who's
mhm,
a colleague of mine, before I left NIA,
mhm.
we helped a little bit, but it was mainly I was cheerleading. She did a study, some initial ones in MS. I haven't seen the one that came out a couple of days ago. You know if it's from Hopkins or somewhere else?
I think from somebody else.
Okay.
Yeah, yeah. So, so even I think there's a recent study in C. elegans. So it looks like when in the staff C. elegans, right? So they actually can improve their longevity. So
Yeah, yeah.
and
Oh, yeah. Yeah, yeah.
right. So because we have one study shows in the mature dendrites, you actually require those a specific metabolite, for example, alpha ketoglutarate.
Ah,
But we find the one with transcription factor just highly enriched in the mature dendrocyte that can regulate alpha ketoglutarate.
mm.
So to promote myelin formation and repair. And then it turns out in the stuff animal like a C. elegans, they can induce alpha ketoglutarate, and
Ah,
they're actually helping the
yeah.
you know
And also during fasting, the ketone bodies go up, like beta hydroxybutyrate.
right.
And it has signaling functions. Actually, there's a lot of evidence coming out. It's this ketone produced by the liver in fasting. It's kind of like an endocrine factor that it can affect, and this is something you can talk about,
Mm-hmm.
but it can affect gene expression by modifying chromatin, these proteins that interact with DNA in the nucleus.
Mm-hmm.
It can actually, there's some evidence there's receptors in the membrane for the ketones.
Mm-hmm.
So and then cancer, obviously, there's a lot of interest in energy metabolism of cancer cells. They have very high metabolic rate. And there's some evidence that in a lot of studies now, and actually in cancer patients, seeing if these people, when they're being treated with chemotherapy or radiation, if they're on some calorie restriction or intermittent fasting, the idea is that it may be easier to kill the cancer cells when you reduce their energy availability.
Yes, I think yeah, I actually I heard from some of the doctor actually they mentioned intermittent fasting can you know motivate mobilize those cells to you know to undergo mitosis. So therefore, a lot of chemo um, therapy, right? So they can target those cells. It shows has a better tumor control.
Yeah,
With intermittent fasting, right? So as I mentioned, right, those metabolites can actually modify the chromatin, particularly like a histone modifying enzymes, right? So they can
yeah.
actually regulate gene expression, right? Yeah, I think this is very interesting aspects, right?
Okay. So you mentioned olig-1 and olig-2. So what are the roles in regulating oligodendrocytes myelination? And is there any role for alterations in these two transcriptional regulators in any brain cancers?
Yeah. So yeah, we find the oligo 1 and 2. So oligo 2 particularly is essential for aldensite formation from the new progenitor cells, and we recently also found is also required for the maturation of aldensite. So yeah, so this actually the study was so initially because I'm the molecular biologist, right? So when we clone the gene, so we don't know what's the function, right? So actually at that time, Dave Roach actually joined Dana-Farber from Andy McMahon's lab in 1998. So he bring the expertise in developmental biology, and then it give us tremendous help to advance our study and to show the origin actually required for the oligodendrogenesis. Yeah. So, but one thing there was a little bit surprise because we at that time we believed the origin expression domain for example, in the spinal cord is a complete overlap with the motor neuron formation domain. So, and when we made a knockout for OLO2, not only blocking the adenosine formation, but also block the motor neuron formation.
Huh, yeah.
Yeah, that's a very surprise finding because the late Tamajaso had been looking for the factor that control motor neuron formation. So he hasn't found one yet. So, but we serendipity found the gene that control motor neuron that control adenosine formation, also control motor neuron formation.
That's really interesting. So I mentioned that the myelination is on neurons with long axons. So is it specific? Is it both upper and lower motor neurons?
So it's a lower motor neurons, right? So
But not upper motor neurons.
I don't think it's upper, right? So only in the lower
Huh.
part. right?
Okay, because I was just thinking maybe there's some. So during development, it may make sense to regulate the in time and temporally turn on at the same time the program that caused stem cells to become either oligodendrocytes or motor neurons, because then I don't know, I'm just thinking out of the box, but that's
Yeah. So yeah, motor neurons is one major neuron, right? They control the one those control a lot of
oh, in that case it would be Schwann cells. Schwann cells.
yes.
Yeah,
So motor neuron actually in the spinal cord part
yeah,
actually is
yeah,
controlled by aldenside, and then
yeah,
go to peripheral nervous system actually wrapped by the Schwann
yeah,
cells.
yeah. Okay, and so talk a little bit, get a little bit into the more into the molecular biology and the regulation of gene expression in the nucleus, and because you've been looking a lot at some of the and discovering new proteins or genes and proteins that regulate gene expression. So this is very important because environmental factors can affect gene expression through epigenetic mechanisms. And so when we think about whether it's normal development and a health throughout life, brain health, health in general, and we think about environmental factors that may affect our health and risk for cancers, then these epigenetic mechanisms become very important.
Yes. So yeah, when I have my independent faculty position at UT Southwestern Medical Center, so I was there for 11 years.
Yeah.
So I mainly focus on the downstream target gene regulated by, for example, oligo 1 and 2. So because at that time, very little is known about the transcriptional regulation of aldendrogenesis. So yeah, we identify many factors actually had not been known before. They have a very important role in the aldenside differentiation and maturation. So yeah, for example, we identify so the O2 can be the upstream regulator for some of the key transcription regulators, for example, SOX10 and ANKase 2.2. And in addition to that, we also found O2 can regulate some other new factors we identify, for example, zinc finger 4A8, one of the zinc finger protein, and also the ZAP2, for example, additional transcription factors. So it turns out there's so many transcription factors involved in the oligodendrogenesis. So I think but if you mutate each individual one, it can cause the defect in the differentiation. So therefore, to push the aldenside lineage, you need to has a lot of factors to safeguard this, the lineage fidelity and also progression, because aldenside precursors are highly plastic. They can be induced to become astrocytes and also to maybe other cell types. So the environmental effect is very strong. So they need to be recruit a lot of factors to make sure they actually only go to the myelinating aldenside.
So overall, then, what you did initially, you discovered OLIG2, and then your subsequent work is showing that it's kind of a master regulator of other transcription factors, and then yeah, so then it gets really complex in determining kind of the overall outcome of any whatever signaling pathway upstream of OLIG2, and are there factors or mechanisms that impinge between OLIG2 and the other transcription factors, and so on. So very quickly it gets highly complex. How do you go about, is a lot of your work, do you do both work in cultured cells and mice, or just
Yeah, we do both.
you do both?
Oh yeah, so for example, we were looking for the initiation of aldenside differentiation. So we culture the ordinary, precursor cells, and then induce them to become differentiation. And then we can do the profiling, and then to looking for the gene expression change, right?
Yeah.
So that's why we actually found out Olo2 actually can work with a chromatin remodeler, like a sweat sniff complex mediated by the BRG1. So Olo2 actually is kind of function like a pioneer factor, so they can occupy the specific location in the genome. So maybe linked to the lineage gene, and then later on bring in the chromatin remodeling complex, like for example, ATP-dependent chromatin remodeler, and to open up the chromatin structure to drive the downstream gene expression.
Wait a minute. So is OLIG2 can it be both a negative and positive regulator of downstream transcription factors?
Yes. So it depends on what kind of cofactor work with the oligo2, right? So yeah, so we found the oligo2 can interact with some different factors, has a different effects, can be positive and negative regulation of the downstream genes.
So for the listeners and viewers, so this quickly gets very complex at the molecular level.
Mm-hmm.
However, this work is very important in identifying potential targets for drug development. And, looking through your publications, you had you at least can, yeah, I think you were the senior author. You had a paper in Cell describing a chemical that's it's called ESI1.
Yes, I got
And
right. Yeah.
you mentioned multiple sclerosis, where the myelination, these oligodendrocytes wrapping around the axons, it kind of degenerates, and the axons actually may degenerate. And actually, I had a podcast on multiple sclerosis, and there's a couple different kinds. One is called remitting relapsing, and then there's one that's kind of more progressive. And there are models, animal models for this. So can you talk about that Cell paper, because it's one example, and you may have more related to cancer, but it's one example of going from nitty-gritty molecular biology and cell culture to the clinic.
Yeah. So
potentially to the clinic. So
So in the MS, so we actually collaborate with the Blue Stripes lab to get the MS sample. So we actually found out actually other people also report there are many of the odensite present within the lesion, but those odensite cannot produce myelin sheaths. So the cells there, however, they cannot do anything for myelin genesis. And when we look at those oligodendrocytes, because we have been working on the epigenetic regulation, we know those histone mark, activating histone marks and also repressive histone marks. So we find those oligodendrocytes within the lesion, they are epigenetic silenced because they have they lost those activating histone mark and gain of the repressive histone mark. So therefore, they are in the epigenetic silencing state. So
the idea is to inhibit the inhibitory factor that's preventing the turning on of certain genes, and therefore the ability of these kind of dormant oligodendrocytes to start myelinating again.
Exactly. So yeah, therefore we designed a screen to find a compound that can reverse this epigenetic silencing to rejuvenate the odensite capacity to promote myelin production.
And this chemical, I looked it up quickly. It has or did I look it up? Does it have anything to do with cancer therapy?
So so this actually was initially identified for targeting cancers.
Yeah.
So prevent the cell proliferation. So they're kind of interesting, right? So so we recently found out a lot of compounds they can promote differentiation, but they also have another function to block the proliferation of a cancer cell. It's kind of like a one stone to kill two birds. We can help the regeneration and also inhibit cancer growth.
Now, Is it established that certain kinds of brain cancer arise from, so I mentioned Mahendra Rao, and
Yeah.
so he contributed to the idea that there are, as brain development occurs, there's restriction in the in which kinds of cells certain stem cells can give rise to. So there's glia-restricted precursors or neuron-restricted precursors. Oligodendrocyte may be restricted precursors or astrocyte. And in the cancer field, we're getting a lot of topics, but I don't know if you've come across, you probably have, Justin Lathia
Yes, yes.
at Cleveland Clinic. He was a graduate student with me.
Oh, really? I
Yeah,
see.
he
Yeah.
worked on neural stem cells
Yes, yes.
in my lab, and then he was interested in cancer, so he went to Cleveland Clinic.
I see. I see.
But
Yeah.
but he talked about there's a lot of work on trying to identify cancer stem cells
Mhm.
in tumors. A lot of times the tumors, when you treat them with these DNA damaging chemotherapy drugs or radiation, the tumor shrinks. But it's become clear that there are certain subset of cells in that tumor that are resistant to the treatments, and they're, I guess, kind of often referred to as cancer stem cells. So then they come back, the cancer recurs, and this is a big problem with glioblastoma for sure.
Yes, yes. So we actually found all the two. So they express very early on. So prior to odensite precursor formation. So yeah, it's a very interesting. So a lot of study actually demonstrate the OLO2 function like stainless maintenance factors. So a lot of glial progenitor cells. So you know, well, like for example, in the gliomas, so those precursor cells they divide actually has a OLO2 expression. So they kind of like a more like glial progenitor cell, just like Mahendra Rao mentioned, right? So
Yeah.
glial progenitor. So before they decide to become oligodendrocytes or astrocytes, so those are actually highly proliferate and they can be you know you know they can be susceptible to oncogenic um, activation to from tumors. So depending on what signaling, right, they can become astrocytoma and oligodendrogliomas.
So if you have some drug that blocks these maybe treatment resistant progenitor cells, stem cells, uh, then maybe like the idea would be to administer the drug beginning, I don't know, I'm just making something up, beginning a week before the person gets the chemo or radiation and continue the treatment with the drug through that time period and then maybe discontinue. The idea being that it might not be good to inhibit these pathways chronically because they have normal functions, but maybe it's okay to inhibit them transiently during this critical window and thereby enhance the likelihood of no recurrence of the cancer.
Yeah. So yeah, particularly for example, for the patient they, after standard care like radiation and chemo. So and then very often those tumor can come back
Yeah.
from the stem cell. And that, right? So that's very important to get the compound which can target these stemness, right, to prevent their further growth, right?
Yeah.
So that's one idea people actually try to target those therapy resistant tumors, right, to block the stem cell proliferations,
Uh-huh.
right?
Okay. Let's see. Right. Can you talk a little bit about, you mentioned the histones. Talk a little bit more about the chromatin and how and what the histones are and how their modification by small molecules like whatever, acetylation or I guess methylation, how that is working. This is a big area now.
Yes, I mean, it's very complicated because the histone modification, for example, acetylation can be regulated by many factors. For example, histone acetyltransferase and also histone deacetylase, like HDACs. There are many of the HDACs, right from 1 to 11. So, and in general, so we found the drug we identify actually can enhance the histone acetylation on the K27 site, so that can motivate or activate the gene expression, and also suppress the histone repressive mark, for example, H3K27 trimethylation and also H3K9 trimethylation. So therefore, if we can enhance the activating histone and then repress the repressive histone status, right? So we can actually, reactivate the gene expression. Because I mentioned in the mature oligodendrocyte, usually you have very low level of gene expression. They pretty much in the dormant or silent state. So particularly under disease environment, right? So a lot of environmental factor actually suppress the gene expression too. So therefore, so we can if we can rejuvenate the gene expression to let them to restart the program to produce a myelin gene, that can actually help to promote myelin sheath formations.
Yeah, that's
Yeah.
very exciting. And then, right, this, for those of you who don't know, so when Dr. Liu mentioned K, what he's referring to is an amino acid called lysine. So proteins are strings of amino acids, and I'm not sure evolutionarily or how this came to be that the lysine is so important, but it can be, there's these enzymes that can either add or remove acetyl groups. For example, people may have heard of acetyl coenzyme A, or methyl groups, I guess. But this lysine amino acid seems to be so critical. I don't know. Do you know if it's has to do with? You'd think it would have to do with something about the positioning of the amino acid within the chain, because these proteins, whether it's histones or other proteins, they have like a three-dimensional structure. And yeah, I don't know. Are they lysines more likely to be exposed on the outside of a protein?
Yeah, so those modifications happen to be on the histone tail. So histone, you have those optimal, right? So from the nucleosome.
Oh,
And
I
then
see.
those are on the tail. So the modification, right, on those histone tail can actually alter the nucleosome structure. For example, acetylation can lead to more looser of the nucleosome, and then so therefore the factor, other transcription factor can come in to activate the gene expression, and also can interact with other the chromatin reader. So, so, and then they can blow in the chromatin remodeling factors. So they can recognize this, the lysine acetylation, and then they can bind and then start to modify and remodeling the chromosomes.
Yeah.
Yeah.
We did a lot of work on a protein deacetylase that's in mitochondria. It's called SIR3, SIR2 and 3.
Mm-hmm.
And the evidence from other labs who kind of try to identify using various ways the protein targets, that is the proteins on which this enzyme acts. And there's hundreds of them, at least several hundred of them. And yeah, it's still kind of interesting to me during evolution why the lysine, because it's not clear to me that the location of the lysine in all of these proteins is like at a highly exposed region. So I don't know.
Yeah. So yeah, this kind of fascinating, right? So David Ellis, right? The late David Ellis, they actually identify those histone code. They call histone codes, right? So you have a different lysine position get modified it, and then actually to totally change the gene expression patterns.
Yeah. Yeah.
Yeah, so this you mentioned about SIR2 is also highly expressed in the oligodendrocytes, so has a role in terms of oligodendrocyte maturation and myelination as well, right?
And energy metabolism and survival and vulnerability to damage and degeneration. A lot of the targets of this SIRT3 in the mitochondria, some of them are in electron transport chain proteins. One of them is superoxide dismutase 2, a very important antioxidant enzyme.
Mm-hmm.
So the SIRT3 increases the activity of that antioxidant enzyme. So it has an antioxidant function. Another one is a protein involved in programmed cell death, apoptosis. That's protein involved in the formation of pores in the mitochondrial membrane that can be a trigger. So there's many ways that, and we actually found using SIRT3 knockout mice and electrophysiology and other methods that somehow mitochondrial SIRT3 plays an important role in synaptic function. Yeah. Okay, let's see. Talk about a few of the other genes. So BRG1, this is one you've worked on a lot. What does BRG stand for and what's known about it?
I see. So BRG is called bromodomain protein. It's one of the key regulator in the Schwann-Smith complex. It's one of the key subunits. So it's a ATP-dependent complement model complex.
Ah, I
Yeah.
see.
So it's very important not only for the oligodendrocyte differentiation, also for neuronal development as well. So
And
it
what
looks
about
like
in cancer too?
so in the cancer too, yeah. So their study actually shows the BRG the complement remodeling factor actually get mutated, so they can drive the tumor growth. One of the subunit called L1D in within this complex, there's a high instance of mutation on that subunit, so that can lead to cancer formation.
Oh,
Right.
right.
And
And there at Cincinnati, so you're a key player in the cancer research program up there. Do you have good oncologists there, clinical people you interact with? Do you have any translational projects or
yes.
kind of a cancer therapy development program?
Yes. So actually, my lab is embedded within the clinicians lab. So just my neighbor, they're all clinicians.
No.
So so we collaborate a lot. For example, when I first came to Cincinnati Children's, right? So we actually do the genomic profiling for the tumor sample from kids. So to identify the potential faulty gene that can contribute to the cancer formations, right? So yeah, one of genetics we find out it's very interesting. So we find one of the homozygous mutation in the tumor tissue, not in the normal tissue. And then when we mutate that gene in the mouse, they can form tumor 100%. The tumor we call megaloblastomas, right? So it's one of the most common malignant childhood tumors, right?
And right, so you started out the collaborations by the neurosurgeons when they do the surgery to remove the tumor, then they give you the tissue.
Yes, yes.
And you ask how is gene expression or whatever histone modification in cells in the tumor different than normal?
Yes, yes.
So
So
what do you use as control? Just normal brain tissue from some source or?
yeah, so sometimes when surgeon when they do the surgery, sometimes they always because they have to particularly one type of tumor like for DIPG or diffuse infiltrated punctate glioma, they are deep in the brain. Sometimes in order to reach to that cancer, you have to
uh,
remove a part of a normal tissue. So there actually we usually use them as a control. Yeah. So one thing I want to mention is in particularly in the children, so there's a specific histone mutation called H3K27M, lysine to methionine mutation. So that leads to a very aggressive tumor known as DIPG or DMG, diffuse midline gliomas. So because the tumor location is very hard to do surgery, so and currently there's no effective treatment, including radiation and chemo, right? So I mean the lifespan for those patients after diagnosis usually within a year. So it's a very aggressive tumor, right?
yeah.
Right. So they actually relate to the K27M mutation. Actually, they cannot be modified anymore. And then this methionine mutation on the lysine 27 position can inhibit another enzyme called polycomb group enzyme PRC2. So therefore, the PRC2 cannot actually add in any methyl group on these lysine residues.
And what about gene therapy approaches to treating any of these? So you have mouse models of brain cancers. Do you implant the cells in the brain, or do you put them under the skin, or both? Or
So we implant them to the brain, right?
right.
So those brain tumor cells, they hardly grow on the skin for some
Ah,
reason. So they love to be
interesting.
yeah in the brain,
So
right? So
there growth factors, or there's factors in the brain that enable them to grow, but the skin kind of, are there inhibitory factors produced by skin cells? That would be interesting to know.
yeah, I think that's a very good question. I think most likely probably the microenvironment are different because
Yeah.
the tumor cell they can actually interact with normal brain cells, for example, neurons. So they can form even form the synapse
Uh,
between neuron and the tumor cells. There's a very recent study by the Michel Mange group that actually show those neuron activity regulated tumor growth.
right. So if you knock out the, say, glutamate receptor in the cancer cell, does that inhibit their growth?
So I cannot recall whether there's any impact. I think I need to find out. So at least if you're blocking the neuronal activity, for example, if you knock out neuroligin 3, which can bind to neurexin in the neurons, so they actually can prevent tumor growth, and also other factors, for example, BDNF track B signaling pathways.
Yeah.
And so
Yeah.
this has been shown, right? So I think it's a very good question. So I don't know if somebody has knocked out the glutamate receptors and in the tumor cells and then see. I think at least some studies show the GABAergic neuron. So if you blocking the GABAergic signal pathway, actually has an impact on the tumor growth.
Yeah.
Yeah.
It seems like it'd be a pretty straightforward experiment to do, right? If you have cultured tumor cells and just whatever, use RNA interference, for example, to knock out the glutamate receptor and then transplant them. And it's tricky though, using drugs that block glutamate receptors, because your brain functions on glutamate, right? I mean, and so it's kind of tricky business, because if you block glutamate receptors too much, your brain doesn't function well. Your cognition is impaired, and yeah.
Yeah, exactly. So probably it's because the glutamate receptor is widely expressed in many of the cells, right? So it's very hard to treat them with the inhibitors.
Yeah, yeah. Okay. And you have any other ongoing studies that you want to talk about? So let's talk a little bit to finish up about your
Mhm.
actual lab. So how big is your group, and how do you manage them?
I see. So we have like a medium-sized group. We have 10 people in my lab. So half of them actually interested in the organ myelination and repair. So, and also help them actually work on the brain tumor projects. So,
Yeah.
yeah, recently for brain tumor project, we are very interested in the immune therapy. For example, based on very unique feature of for example, specific tumor, they have a very unique tumor antigens. So we can design the CAR T to target them uh,
Uh,
to eradicate those tumor cells, right?
but what about in the, yeah, it's interesting in the brain.
Mm.
For a long time, it was thought that T cells, which by the way are the cells that when you get a COVID vaccine or flu vaccine, right? Those are the cells that are turned on and target the viruses. So for there's this blood-brain barrier, right? That not everything can move through, and for a long time it was thought that there are uh, no lymphocytes, no T cells, for example, in the brain. But there's some evidence. It's pretty interesting. There are little pockets of T cells like are sitting what's called the choroid plexus under the brain, right by the blood-brain barrier, and in certain conditions they can move into the brain. So is the idea to yeah, I guess so the CAR T therapy that's used for certain blood cell cancers, there you've got the circulating blood cells and the kind of the T cells are circulating with them. But to target brain cells which are in the brain, are these cells, if you yeah, talk about kind of the technical approach are the kind of the hurdles that have to be crossed.
I see. So I think recently, people have been trying to just inject the CAR T cell directly to the brain. So and then it looks like it can eradicate the tumor cells and also improve the neurological functions.
Ah,
So that's kind of amazing. So most people believe like T cell are kind of like a immuno privilege, right? The brain are the immune privileges, right? So now actually, indeed, you actually can put a T cell in and then to target the tumor cells. I think people also trying the NK cells, right? So which can there's a recent study you can modify the NK cells and then this was done in the animal directly inject to the brain. Uh, of the mice carry the glioblastoma. So actually, it turns out the NK cell can actually target those the GBM cells,
the natural killer
natural killer cells, right?
cell. And so are there are clinical trials going on now with these approaches for brain cancers?
Yes, there are many clinical trials actually using those immune therapy, right? The T cell based immune therapies.
Yeah.
Right.
Okay, that'll be. I remember it's been at least 10 years ago that there's a group at Duke that they were, what were they doing? They were injecting some virus into
Yeah, herpes, right? So
yeah. Which virus?
I think it's a herpes virus.
Yeah, but there was another one, like not rabies, but okay. But anyway, the idea is that you you cause a reaction, immune reaction to the virus. So you
Mm.
inject the virus in the tumor, and then the immune system is maybe its local immune system is recognizing the virus
Hmm.
and causing a lot of inflammation and these cells, like the microglia, for example, they release oxygen-free radicals. They release uh, well, pro-inflammatory cytokines, some of them which could maybe damage these, like tumor necrosis factor, right? Its name, TNF, tumor necrosis factor, comes from the fact that I guess the early discovery was that, this inflammatory cytokine can kill cancer cells.
Yes.
Yeah, so
So that's true. So those are herpes virus. The virus actually can induce to activate those immune cells to release those tumor killing cytokines.
Yeah.
Yeah, that's the idea. So I think we can mobilize our immune cells to target those tumor cells. And in particular, for example, now you have those immune checkpoint inhibitors, so you can block the tumor cell. They are very smart. They have those seals, right? You can break the seals, so therefore the T cell or immune cell can recognize those tumor cells and then to kill them, right?
Yeah.
Yeah. So yeah, for my other study for the regeneration, so we are recently also very interested in the Alzheimer's disease. So there's one study actually show myelin loss can trigger the aggravate the disease phenotype in the Alzheimer's disease, and then they even show the aldensite can actually produce A beta to contribute to the A beta formation. So because we have those, we can manipulate the aldensite, the number, and then we're actually looking into how the aldensite, whether we can, by, manipulate the aldensite activity, right, to, reduce A betas. Yeah. So we do have some preliminary data to show our drug actually can reduce some of the aberrant neuron activities in the AD mice.
Ah,
Yeah.
we one of the last studies that was done in my lab before I retired was Pei-Su Zhang with collaboration from others at the NIA, but it was mainly her. There's this accumulating evidence that glial cells can undergo senescence
Yes.
during aging, and maybe more so and acquire an inflammatory bad phenotype in maybe Alzheimer's. And so Pei-Su provided evidence that that oligodendrocyte progenitor cells undergo senescence in Alzheimer's mouse model and in AD, human AD brain tissue.
Mm-hmm.
So that's another potential thing that targeting these oligodendrocytes.
Exactly. Yeah. So we are also try to find a way to reverse those senescence, right? So there's another factor that prevent them to form myelin.
Yeah.
Yeah.
So a lot of exciting things going on regarding the oligodendrocytes and the stem cells in the brain in relation to this basic science, understanding normal brain development, plasticity in the adult, and then what's going wrong in a range of diseases,
Exactly. Yeah.
demyelinating diseases,
Mhm.
cancers, maybe aging Alzheimer's disease.
Right. And also a lot of developmental disorders. So for example, autism, one of the most high risk gene mutation we know as CHTA. It's a completely modeling factors. Chldren.
Oh.
So mutation in that factor actually has a very profound impact on the white matter myelin formations. So yeah, this actually we find this a completely modeler as one of the target for all2. So therefore, when we make a knockout of this gene in neuron, there's no so much impact. But when we knock out in this gene in the aldensite, it completely wipe out the aldensite formations. So therefore, actually, it's consistent in the field. People actually believe there's a profound myelin loss in many of the autism subjects, right? So, and also, yeah,
Go
a lot
ahead.
of a lot of factors actually can cause a developmental delay, particularly for white matter development. So that has a profound impact at the later in the adulthood in terms of their cognitive functions.
Yeah, and myelination, particularly in the forebrain, the most highly evolved region, occurs very late in humans. And there's this idea and evidence that since those brain regions are very important in decision making and impulse control, that like if there's a delay in myelination, then looking at this kind of simplistically, someone in their 20s or 30s may be as impulsive as a teenager, or make bad decisions.
Yeah, it's impossible. Yeah, so a lot of studies actually show the early impact has long-lasting effects. So they call it like a epigenetic memories. So
Yeah, yeah.
yeah, they can be have a long-lasting effect in the later function of the brain.
And these epigenetic changes may occur even before birth.
That's true. That's true. Exactly. So, for example, the environmental factors like alcohol, right, sometimes actually do change the chromatin structures, right? So, yeah, that's
Or even Richard, even obesity and diabetes during pregnancy, that increases the risk for autism. And there's some work, quite a bit of work actually going on that obesity in a parent can result in epigenetic changes in the child or offspring. A lot of this is in mice, but yeah, so it's a very important area.
yes, yes, yeah. Obesity also been shown in mice, right, can impact the myelin formation. So, yeah,
Yeah, yeah, yeah. Okay, a lot of exciting things to explore, and I
yes,
enjoyed you taking the time to talk. I've learned a lot, and I will put in the description section links to some review articles to your website, and uh, which people, if they want to follow up and kind of in a more slow manner, if they don't have any background in this, understand, why your work is so important for so many aspects of brain research.
yes. Thank you. Yeah, it's a very broad area, so there's so many things unknown. So, we still, very interesting in many factor things
Yeah.
around this area. Right.
Okay, Richard. Thanks a lot. Goodbye.
Oh, thank you so much. Thank you very much for having me. Right.